list of terms, and these are all of the vocabulary words and gene names that I consider important that you would be tested on for that lecture. So this is one of the things, like the drawing a boundary of what's important, what's not important in a big class with biology and lots of factoids here and there. So this will help focus it down. If it's on that study guide as a term, it's fair game for a test. If it's just a gene I mentioned in passing and it's not listed there, then don't feel like you have to memorize that particular situation. There's going to be about usually 4 or so questions, and they're deliberately open-ended questions. They're starting points for how to study this material to kind of get you thinking about it, because we don't want to just come in here and memorize stuff, right? We want to actually learn some concepts and grapple with stuff. And so take these questions as a starting point and reflect back on the lecture, what we went through that day, see if you can draw out some of the concepts based on that. And then I've also recently added just some statements or bullets about what the major concepts are for each lecture. So that's all to kind of trigger your self-guided study. Okay. Then we have the schedule of topics here. And you can look at this. Some days we're going to do what are called in-class exercises. And that means that I stop blabbering on up here and I give a worksheet out, and you guys have a chance to work together on a concept relevant to developmental biology. And then we'll kind of come back together as a group and see, how far you got, what you discovered. And I've done this in the past and people really like it. It seems to be a good way to have a little bit more engagement in the class and just to break things up a little bit. And especially with 2 hours every day and a sunny summer morning, it's not the worst thing. So you do need to be in class in order to do these. So come to class and you'll get a few points for doing that. If you sort of irrevocably cannot be in class for some unimaginable reason that's more important, these will be posted on the website afterward, and you can do them on your own for half credit. So they're going to be worth 5 points, so you could do 2.5 points because you missed the discussion part. Then our exams are always scheduled for Tuesdays. So that way I'll have a nice long break of Friday through Monday, Tuesday to learn the most recent material, because the pace in summer kind of keeps going. So I didn't want to set it up so you'd be taught something one day and an exam the very next day. So you'll always have the full entire time to do the exam, like both class periods essentially back to back. And believe me, that will be more than enough time. I expect you would be able to do it in 40 minutes, most people can. We'll always have a review session before each exam, and during that time you can ask any questions. You can ask questions from the study guide that maybe you or your group weren't quite sure what I was looking for or thinking about with that particular study, that question. Okay, then grading. So the labs work 25% of the grade, hence it's important to go to lab. And then from the lecture, we're going to have 3 exams. And this is a little bit different than the spring, which they have 4 exams, but I've just broken it into bigger chunks. And they'll be worth 125 points. We'll have a comprehensive final, and that I'll talk more about when we get closer to these in-class exercises, and then the 25% for your lab grade. So usually, well, in fact, I've never actually really curved this class. There might be a very, very slight curve, but not much of one. And actually, that's good news for you, because the reason I don't do that is that I don't need to. Everyone does well enough that the average comes out to be right in the mid-70s. So that works out well. And you can read through the rest of these points. I think they're pretty standard in terms of coursework here. So any questions? All right, then let's go ahead and get started. so for the very first lecture, it's of course just an introduction to kind of what we're talking about and to the idea that genes play a really critical role in developmental biology. And I thought I would actually start out with just an introduction of me. So like other faculty here at CSU, I run a research lab, and I am a developmental biologist, which is fun for me to teach this class because I love the subject, and it sort of is, something I enjoy learning about in any case, so it's fun for me to get to share that with people who are at a good stage in their careers to learn about this. So what we work on in the lab is this fish called zebrafish, and actually we work on the embryos, as you might think, for developmental biology. And we study the development of the heart. So one of the major birth defects that humans can get is in the way that the heart is constructed within the embryo. And genes play a huge role in this, and so we're trying to figure out, just from a basic biology standpoint and from a birth defect standpoint, what are the important genes to build a heart. And believe it or not, the zebrafish and the human have a lot of similarities in the way they accomplish this goal, in the way that they construct a heart from cells. And so you can look at these major landmarks and recognize the similarities, and the genes that are important for each step are very similar between the two species. So it's not just me, but a lot of people around the world are working on zebrafish as a model for organ development, including the heart and other things. So this up here in the corner, this shows you a zebrafish heart with 2 chambers, and it's actually labeled with a marker called green fluorescent protein that we'll hear a lot about, of course. So one of the nice things about zebrafish is they're transparent, so you can watch their hearts in the live embryo and see how they're contracting from the very earliest stages. So just when this tube actually gets constructed and made and starts beating, from the very first heartbeat, you can see this in the live embryo without any dissection. So now think about that. You could never do that in a mouse because the mouse is in a placenta and it's inside the mother and you just don't have access. So this is really one of the really serendipitous, beautiful features of zebrafish, is that you can have this really critical early access to watching the process unfold. And of course, if there's any defects, you can see that as well. So this has applications to human syndromes, such as a syndrome known as Holt-Oram syndrome. And this turns out to be a mutation in a transcription factor known as TBX5. And in humans, what you see is defects specifically in the formation of arms. So you can see the patterning of these arms, the construction of the different elements has gone wrong. And then they also have congenital heart defects. So lo and behold, when you have a mutation in TBX5 in zebrafish, you see the absence of these pectoral fins. They're just totally gone. And you also see a cardiac defect where the heart does not form normally, and the embryos end up dying because of this malformed heart. So 2 totally different tissues, the forelimbs and the heart, and both the fish and the human have an exact parallel in the defects within these 2 structures. So we can learn a lot about what TBX5 really does inside the cell in zebrafish, and then translate that over to humans and see whether or not it's doing exactly the same thing, and there's bound to be some similarities. Okay, so developmental biology, starting from the very biggest picture, what are we talking about in this field? So in many cases, organisms are going to start out as a fairly amorphous blob of cells, if you will. So they all start out as a single cell that divides a couple of times. There's no real head or tail at this point. It's hard to define what's the front side, what's the back side. And then we'll go through this period of embryogenesis, the building of the embryo. And look at what we get out on the other side. An amazingly complex individual with patterning, with organs, with a brain, with all sorts of differentiated cell types. And the development of biology is the process of how this happens. How do you get from A to B? And for me, that's always been a really, really interesting question as a biologist to think about, how is this controlled? What are the genes that are needed? What do you need besides genes, if anything? And how do you actually go about making such a complicated product at the end? So as we go through this course, we're going to kind of break it up into parts and consider different aspects of developmental biology. Just to get started, we'll talk about some of the major stages and things that we see. We'll talk about patterning, which means anterior, posterior, dorsal, ventral, left and right, how we decide that. Organogenesis, the construction of organs. Post-embryonic development, so this would be more like puberty, how reaching sexual maturity. And then at the end we'll talk a little bit about plants too, because plants are organisms and they definitely have some developmental biology that they have to go through in the course of their life history. So with developmental biology, it's actually a great time to be taking this course and a great time to be a scientist in the field, because there's been so many recent advances with genome technology and genome sequencing that we're now learning the answers to some of these questions that have been around in the field for 100 years or more. So I think you'll see that kind of coming out as one of the themes, that we're now at a place where we're making really rapid advances in this particular field. Okay, so here's just a kind of a general concept slide where we look at these different stages. So as we start out with an egg and a sperm, when they are joined together to create the one-celled organism, this new individual is termed the zygote. So that zygote, and just for those events of fertilization to be successful, that already takes a lot of specializations within the species. So we want sperm of species A to fertilize the eggs of species A and not of species B. So 2 or 3 different species of sea urchins living in the same environment in the ocean, there needs to be some mechanism there so that the correct sperm fertilizes the correct eggs and the species can be propagated. So we'll learn about some of the mechanisms behind fertilization. Then once we have a successfully fertilized egg, that thing needs to go through many, many rounds of mitoses to start to create the multicellular organism. And so that also has some kind of rules or principles that are guiding that process so that you can get out something that has some structure to it. It's not just a random division of cells. It has to be done in some sort of a planned way. So we'll look at what's happening there. That's the stage during this cleavage where that first big cell gets divided up into many daughter cells. Then, many organisms, most animals, go through this process known as gastrulation. And this is a very dramatic time in the life of the embryo, because what is a pile of cells now goes through this massive rearrangement. And you'll literally see whole tissues of cells moving to new locations in the embryo. And it's almost as if the embryo turns itself inside out. It's that dramatic of a rearrangement. And so what this is going to do is, in the end, to set up 3 different layers that will be the foundational layers that all other organs and tissues come from. And I'll just tell you the names so you'll recognize them when you hear them again. It's mesoderm, endoderm, and ectoderm. And that's what we'll get at the end of gastrulation. And then we'll tie in from those tissues what happens to each of them as time goes by. Then we have a whole period where we're building organs, such as the heart, the liver, the limbs. So each of these has a particular need in that you have to construct the pattern and the functionality and the specialized cell types in there. So it's almost a microcosm of developmental biology happening in that little area of the embryo at the time. Then we get to the boring stage where we've done all the interesting bits for the embryology and the thing just needs to grow. But then beyond that, there are questions of sexual maturity. What do you need for that organism to become capable of producing sperms and eggs so that the next generation can ensue? So there's an interesting observation that biologists have made through the years, that there is a period of development. If you compare different species, there's this one period of development where all the embryos from various different species really look remarkably alike. And that's this period right here. So here we have a frog, a fish, chicken, mouse, and human. And all these embryos have a certain similarity to each other, and that they have a recognizable head and tail. They are all kind of making organs at this point, and just morphologically, they're reasonably similar. In contrast to that, if you look earlier, say if you look at the stage where it's a one-celled organism, and we're talking about the fertilized egg, well, here there are huge differences. The chick egg is massive. We can see a chick egg, right? We've eaten those for breakfast. They're huge. The human egg is tiny. You can't see it with the naked eye. Frog egg, zebrafish egg are kind of in the middle. So there's a huge discrepancy in what things look like earlier than this stage. And then when we get to the final adult organism, they're very, very different morphological creatures. So, why is it that there's this stage at all where they look somewhat similar? And, part of the reason for that is that all of these organisms have, to some degree, similar goals. They need to establish a body plan where they have a head and a tail and a left and a right and a back and a front. They all need to make organs, and they all need to go through cell differentiation. So that may be one reason that contributes to at least this similarity. So what about plants? So plants are a little bit different. They have two different parts to their life cycle. Part of their life cycle, the individual will be composed of haploid cells. So one in ploidy. And then part of their life, they'll be composed of diploid cells, two in ploidy. But they still go through a lot of these same stages. So they have a sperm and an egg that need to fertilize. They have a cleavage stage. They don't actually have a gastrulation stage. Could anybody think of why that might be?
Stomachs.
Sorry?
They don't have stomachs.
They don't have stomachs, so it's something more to do with the cells. That's something that the cells have unique into plants.
Cell walls.
Cell wall. Right, so if you have a rigid cell wall surrounding your membrane, that's not going to be that easy for that cell to go through a migratory phase. So they're much more limited in the way that these cells and tissues can move around. They do make organs. Leaves, shoots, and roots are all considered plant organs, so they do go through organogenesis. And then they do have growth and maturation things to consider in the life cycle. Okay, here's one of our first sort of major concepts to throw at you, the idea of cell fate. And initially, the zygote is kind of a generic round cell, but as time goes by and more cells are created in these organisms, cells begin to be specialized for different tasks. For example, you could have a muscle cell, or a neuron, or an endocrine cell. Each of those types of tasks and lives would be considered a cell fate, so a specialized differentiated phenotype for that cell. And one question is, how do we get from this more or less generic zygote or early multicellular embryo to an embryo with cells that have these combinations of very specific cell fates? And how do the cells know, well, I'm supposed to become a neuron? How do they get to that process? And there are a couple of different ideas about how this might happen. The first idea is that this process could unfold like a lineage. So it's going to be based on what was the pattern of cell division within the embryo. Maybe these one cells in this one region always become the gut, and a different set of cells that are descended from a different path, they always become something else. And so it's very planned right from the beginning exactly what daughter cell would give rise to what fate. That would work. And in fact, that does happen for some organisms. A different way to do it would be more of a based on the local environment and the interactions with other cells in the neighborhood. So this would be like, a group of people who all live in a neighborhood getting together for a cleanup day, and they decide, well, you're going to do that, you're going to do that. John, he's not here this day, so we have to get someone else to do that. And so based on their interaction with each other, decisions are made about eventually who is going to do exactly what task for that particular group of people. And that also would work, and we do see this happening too in organisms. And depending on what organism it is, you get more of one or more of the other way of pulling this off. Another theme that we'll see is that as the embryo grows and goes along, the potential, the choices for any given cell about what final cell fate it will adopt becomes increasingly more restricted. So here I have the zygote up at the top, and by definition, this is the mother cell for all other cells that the organism will ever have, right? All of our cells can track back their lineage to the zygote in some way. So I've represented here the full range of cell fates by colored boxes of every possible color, representing every possible cell fate for that cell. It has a potential to give rise to every type of daughter cell that there is in the organism. Then we go through some cell divisions here, and now we're looking at one of those daughter cells, the blue guy there. That daughter cell has already become much more restricted in the possible choices available to it. So it may still choose one of, say, 16 fates, but it's eliminated already the other 198 fates that it might have chosen if it had been in a different spot or had different interactions. Then eventually we'll have to go through a period of time, a mysterious window, where the final choice is actually made. I'm going to become a neuron. And so at the outcome of that, we'll get cells that have a more differentiated phenotype. So let's look at some terms to talk about this. Totipotent refers to the zygote, and specifically it refers to the ability to make any and all cell fates within the organism. So the daughter cells of the zygote can make any kind of fate there is in the organism. Toti sounds like total. Then we get pluripotent. And this is in a way pretty similar to totipotent, but it's just a little bit more hedging. Like we're going to make a lot of different cell types, but maybe not every single one. Okay. Then we get critical period of commitment. And once a cell is committed to some particular developmental fate, we call it determined. So not that cell at that particular time, if you look at it in the organism, it may still look like a generic round cell, but internally things have been changing. Genes are turned on and off, and that cell has made a decision, if you will, that it's going to take on a certain fate. So then finally, after all that goes through, we'll eventually get to the point where the cell is showing the structural and functional characteristics of a differentiated cell. So we can term it differentiated at that point. Now, so these are fine to have words to try to describe all this, but in a sense too, these terms are a little bit squishy. When do you think of a cell as being differentiated? It might be when it starts to turn on certain key genes. It might be when it makes cell junctions. It might be when it makes an epithelial surface. It's a little bit fuzzy to say exactly, okay, now instantaneously we should call this cell as differentiated. So just keep that in mind. Oh, and then we also have specified, we'll talk about this, and that's more of an earlier stage along this stepwise commitment to cell fate. Okay, so here are some of the questions that this brings us to. We have cells, they're dividing, they're taking on gradually a more restricted ability to become certain cell types, and eventually they do choose one. But how are they doing this? What are the actual mechanisms? And this is something that I really hope you'll gain from the course. It's not just a memorization of all the different stages that cells go through, but an idea of how it happens. So what are the mechanisms driving these forces? So we're going to talk a lot about genes, and you'll see genes turn on and off at particular times. How do they know how to do that? The organism or the cells may change shape. They may divide or stop dividing. They may kind of keep track of time so they know where they are in the progression of things. They may keep track of position. And to me, this is one of the really exciting and mysterious things about developmental biology, is that somehow this happens, and we now have a great deal of tools to get in there and experiment and figure out what's going on. Okay, so let's look a little bit at specification versus determination. So these are two terms, and they both appear in this, they both apply to this period of time where cells are making decisions about what's going to happen, how they're going to commit to a fate. And so initially, we could say cells are specified if they have the initial information that would commit them to a particular fate. So they may be now capable of differentiating to that cell fate based on the genes they've turned on, their position in the organism. Everything is looking good. But they still, at this early stage, have the ability to change if they get exposed to different signals. So if experimentally you took them out of the organism and you put them in a new location in the organism, or you put them in a petri dish and added some factors, you could still cause those cells to switch and become a different cell fate. So they're malleable. And some people haven't really heard this term, but malleable means changeable. It's like they're clay and they haven't hardened yet, you could still smush it into a different form. So they have some information, they have some patterning, and left alone they do it, but they could still change. Determination is a little bit further down the pathway. Now they're more locked into their fate. And even at this point, if you try to add different factors, move them around, they're not going to change because they've just gotten that much further down the line. So as an example for this, I think of, say, a biology student who wants to become a veterinarian. And I know there's probably nobody in this class who wants to do that. So someone who wanted to go down that career path would, say, come to CSU and start as a biology major. And they start taking all the prerequisites, they get through calculus and physics and all this. So they're starting to be well on their path to becoming a veterinarian. And at this point, they're specified. They may have friends that are art majors that start taking them to museums and showing them cool pictures and talking about the history of art and getting them all excited about that. And it could happen that that student would then change her mind and say, you know, I think I'm going to go for an art career instead of a veterinary career. But if you fast forward now, say 3 or 4 years, and the person has been admitted to veterinary school, and they've gone through all that process and hard work to get there, and they've paid all that money, I don't think that how many art major friends you have, you're going to switch at that point. So you're going to be a lot more locked in. But on still, these are kind of squishy terms. This is a progressive thing that kind of happens as time goes by and events happen. You start to get more and more solidified in your choices. We will be talking about a number of different organisms in the course. And in the field of developmental biology, there's a kind of a limited number of organisms that everybody tends to study. And these are called our model organisms. And the reason we use these is because they have some serendipitous features about them that make them useful as tools to try to discover concepts and principles of developmental biology. So for example, chickens, they have huge eggs. And also the eggs are external to the mother. So you can see them, you can get in there, you can watch how the embryo is developing right before your eyes. Okay, you have to take off the eggshell, but beyond that you can see a lot. Frogs, they also have very large eggs. You can get a ton of them. You can get hundreds of frog eggs to do experiments on. That purple thing over there, that's sea urchin. And we will be working in lab with these. And they're a great model for fertilization. You can really discover a lot about fertilization from studying these experiments. And you can even do manipulations where you take C-Urgent egg and C-Urgent sperm, put them together, and watch what happens. This down here is a worm, a nematode worm called C. elegans. And this has been a really good organism to learn about the really early stages of cell division and the very first cell fates that get established and how this happens. Now with this one, the nematode worm and the famous fruit fly, you can do genetics. And that is huge, right? Because you can get mutants. And every time you have a mutant, you have a handle on a gene that is critical for that process. So if you have a mutant in eye development, well, you have identified a gene that's critical for eye development. However, all of those, so chicken, frog, it's a little bit difficult to do genetics because they have such a long life cycle. Frog, it takes about a year to grow, fertilization of embryos, all the way to adults. So it's a long time in graduate school if you're trying to do genetics with frogs. Compared to 10 days for frogs, or 10 days for flies, and 3 days for worms. Then we have the lovely zebrafish, is a vertebrate, so nice parallels with human development and quick, very rapid development, tons of eggs, and mice. So the tools for mice have improved so much that there's this almost like a threat that mice are going to dominate the entire scene of development of biology because they're just that awesome. And they're, of course, a mammal, so not only a vertebrate, but a mammal. So we have learned a ton from mice. Okay, so talking about the idea of how a cell decides on a particular cell fate. This tracks back to genes. An early idea on how this might happen is that if you look at the genome of the cell, the entire group of all the chromosomes in the DNA that are within those, that maybe as cells take on different specific cell fates, they would actually lose, physically discard small portions of their genome. And so that's what would make these cells different from each other as you look across the organism. And this turns out